Here now with Professor Nikhil Malvankar of Yale, studying protein nanowires. Huge topic. Let's look at what important discoveries your lab has made regarding protein nanowires and Geobacter. Did I get that right?
Yes, so Geo is Earth, and our lab studies this bacteria called Geobacter, which is one of the most common bacteria in all our soil. And this bacteria can grow without any oxygen like soluble molecules by projecting tiny protein filaments called nanowires. And in 2009, our lab discovered that these nanowires have made up of metal containing molecules called cytochromes. And in 2020, we were able to discover some other nanowires. And this has completely changed our understanding of how this bacteria can respire without anything like oxygen.
That's incredible. That's pretty unusual too, I imagine.
Yeah, it's really the Mother Nature has perfected billions of years of evolution so that they can use these nanowires as a snorkel to send electricity over 100 times the size of a bacteria.
How did these discoveries lead to creating the first synthetic assembly of nanowires outside bacteria?
So these nanowires are very difficult to study with Geobacter because it's a challenging bacteria to grow in the lab. So recently our lab with Yuri Launder was able to genetically engineer bacteria to express the protein which makes these nanowires by the work led by my student Yangshi Gu. And now we can make these nanowires both at large quantity and high purity without using Geobacter. So we can figure out how bacteria can make these nanowires on demand. And these nanowires have a sugar binding domain which bacteria can remove, and that assembles them into nanowires.
What applications could we see from this? So
my dream is if we can charge up our cell phone with these bacteria which are ground beneath our feet. But in the short term, these bacteria are important for many environmental processes such as cleaning up radioactive waste, generating electricity, and understanding how they use these nanowires will allow us to speed up these processes. And just like Geobacter, many bacteria which either consume methane or produce methane also show genes similar to these nanowires. So we are now looking into how can we use that knowledge to control how much methane is released to the atmosphere to combat climate change.
How fast can these developments happen?
So now we have the molecular structures available. So we are now working in the laboratory to understand how electrons move in the system. And once we have the mechanism understood, then we'll be able to harness that knowledge.
So much more to see about protein nanowires. Thank you so much.
Thank you.